everybody, and welcome back to our video series covering all the topics in MMP. We are now on to exam 3 content, which is widely regarded to be the easiest exam. I also think a lot of the information in these set of videos will be very comprehensive, just because I feel like a lot of the material is more or less very surface level. So I think these videos should be really helpful to you as a resource. And today we're going to be going over basic genetics as well as genetic testing. So a few definitions I want to get out of the way first are pharmacogenetics and pharmacogenomics. Pharmacogenetics is the study of how drugs and drug therapy affect a single gene, whereas pharmacogenomics is how a drug or drug therapy will affect the entire genome. So there's about 100,000 genes in humans, and these are stored in chromosomes. We have 23 of them, 22 somatic and one pair of sex chromosomes. Chromosomes are made up in essentially two parts. They have their P arm, which is the smaller of the arms, and their Q arm, which is larger. And in the center, they have something called a centromere, which bind those two arms together. Each pair of chromosomes codes for a single gene. And each pair of these makes up a set of two alleles. An allele is essentially a gene on a specific chromosome. And these can be the same gene, which are defined as homozygous, or different, which are heterozygous. So this right here is a heterozygous gene pair because it codes for a blue eye and a brown eye, whereas these two pairs are homozygous because they're both either brown or they're either blue. And what this is called is these pairs of allele are a genotype. However, these can produce a different phenotype, which is a physical representation. So you can see that heterozygous blue and brown eyes make brown eyes, same with homozygous brown eyes, but homozygous blue eyes code for blue eyes. That's like the physical trait, which is a phenotype. Now for a little bit of definitions. In far nomenclature goes for genes and alleles, this greater than symbol represents a substitution. This is essentially a nucleotide gets switched for something else. If you may remember, codons essentially are made up of adenosine, thymine, guanosine, and cytosine. This means that essentially one of them gets switched for a different nucleotide. Let's say it's a guanine, it could get switched for a cytosine, which could cause a mutation in some cases. If a mutation occurs, it can have some different effects in the body. We have what's called a germline mutation. This is essentially a mutation in a sperm or an egg cell. The entire organism produced will have that mutation. However, there's also somatic mutations. And this is essentially maybe in early development, but not at the level of fertilization. A cell may mutate. You can see if it happens very early on, you can have a whole region of the body with this mutation. However, if this happens later in life, in some cases, let's say like a cancer, it may only affect a certain region of the body. So a few more definitions. A polymorphism is when a genetic mutation, or I shouldn't call it a mutation, a genetic difference is in greater than or equal to 1% of the population, whereas a mutation is a change that occurs in less than 1% of the population. So polymorphism may be in like a group of people that are isolated in a certain area, whereas mutations typically are more rare and more by random chance, not super expressed in the population. Now, mutations can occur, like I mentioned before, through the substitutions. Typically, single nucleotide permutations will occur in about 1 in 1,000 genes, or roughly 3 million per genome. And there's a few definitions that go along with these as well. As you may remember from our unit on RNA, amino acids or codons can be degenerate in the fact that more than one genetic sequence may code for the same amino acid. So if, let's say, a cytosine goes into a guanine, and it's the same amino acid it codes for, this is called the synonymous mutation, and this doesn't actually change the genetic sequence. Missense, on the other hand, will be, let's say, that same guanine goes into an adenine. Let's say this does code for a different amino acid. This is called a missense change, and this could affect protein function potentially. There's another type of this called a nonsense mutation, and this is when our guanine, let's say, goes into that's a bad example. Let's just say an amino acid changes into a stop codon or something that codes for a stop codon. This is called a nonsense mutation, and it will cause the protein to stop early. And essentially, it may be short and essentially have no function because it stopped too early before it could be shaped in the way it needs to be. So how can these mutations affect how genes are transcribed? Changes in our promoter sequence can actually change how often a gene is transcribed. This can be with something like transcription factors binding. So let's say we have a change in the promoter here. This may not be able to bind as well, depending on what that mutation happens. And the way we can actually avoid this is we do have a genetic way around this, because obviously we need our genes to be transcribed. So something that we've evolved to do is have what are called short tandem repeats. So you can see that this early region is the exact same in these genes. And that's so if essentially one of these gets mutated, our promoter and our transcription factors can still bind to our promoter, so the gene can be transcribed. Because the odds of all of these being broken is very low versus the odds of one of them being broken. You may have seen on the previous slide, insertion and deletion reactions may occur as well. These are usually more severe than a substitution in the sense that these can cause what is called a frame shift. Um, if you remember from the RNA unit, codons will essentially be read in pairs of 3. Um, and if something is inserted or deleted, it essentially causes a frame shift or moves the entire reading frame. So you can see, well, an insertion may essentially insert one nucleotide, it can cause like this downstream cascade of different amino acids, which can usually lead to an early stop codon, or in most cases also a dysfunctional protein. And same with deletions. Removing one can also cause the same problem, because if you're reading these in these pairs of 3, then you can cause a lot of problems even just by moving it one place. So far we've been talking about mutations involving a single nucleotide, either that being inserted, deleted, or substituted. Um, there can also be mutations that involve entire like sequences of DNA that are moved around. One of these are inversion. Essentially what happens is genetic material between chromosomes gets switched and flipped. This is a little more rare. A more common one is duplication. Entire regions may be copied over and multiplied to other places. You can see that more here. This is more of a duplication image. Um, and that can cause a lot of things. If it's duplication of an entire protein, that would cause it to be upregulated. But if it's duplication of just a specific sequence and it's paired right at the middle of another sequence, it may prevent a gene from being transcribed. Usually, though, it's the entire protein if that happens, so you'll have like a lot more regulation of that proteins. Um, a little more nomenclature. Whenever something is mutated, it will essentially be with a star and then the mutation number. A letter is also like a subvariant of that mutation. So for example, if these star 1 mutations were to cause a stop codon, these A, B, and C would just be other variants that cause a stop codon early in the CYP2D6 enzyme. So star just means mutation. And usually star 1 is what's referred to as the wild type allele. This is what's normal in most of the population, or at least most frequent in most of the population. There is an exception to this, which I'll get to in a few slides, or possibly in the next lecture, but I will make a note of that. Just know that star 1 is usually your normal wild type and usually functional. And just another definition is haplotypes are essentially sets of alleles which code for genes. So on the previous slide, we had our example of the blue and brown eyes. However, you may also know that like brown eyes is typically linked to brown hair. That would be an example of like a haplotype. Okay, so what we've been leading up to with this is, well, promoter regions can be changed and transcription factors, how much a proteins express. We can also change the amount of metabolism, and not only metabolism, pharmacokinetics, but also pharmacodynamics, how drugs affect the body based on mutations. And that's a lot of what this section of the course is about. So essentially, genetic changes in enzymes, for example, can change how much a drug is broken down, how frequently. And let's say there's something like a transporter on a surface that lets a drug get into the cell. A mutation in that transporter may alter how much of that drug is in the blood or how much gets into the cell. So these are a few examples of kind of how important our genome is for how drugs interact with our body and how our body interacts with drugs. Um, so pharmacogenomics has become a lot more commonplace as like a research topic than pharmacogenetics in recent years at least. We used to look more at pharmacogenetics if we noticed a certain enzyme was breaking down one of our drugs, and you may even see those as black box warnings still. They're still around today. We wanted to focus more heavily on that, but as we mapped more of the genome and got a better understanding for how our genetic sequences work, we were able to look larger picture and see how our entire genome can affect drug metabolism. Something else, just a stat, is 7% of approved medications currently have actionable pharmacogenomics, meaning that 7% of the drugs we have on the market, we can kind of predict how they will work with our body, and what will work better for some people than others. Um, and something else to note, oh, and that these 7% as well are 18% of total prescriptions in the market right now. So I'll write that down. And these numbers may actually pop up on your exam, or maybe at least a quiz. I think we got like one question on this. So it's not like a super heavy topic, but something to be aware of. Um, Even though it's 7% of approved medications have this like actionable potential, those make up a total of 18% of all the current prescriptions being prescribed to people. So you can see that doctors will prescribe these almost 3 times more than other medications. If you do like 18 divided by 7, that's roughly 3 times, just because doctors and prescribers are beginning to realize that since we can map genomes and understand how these drugs work a little bit more, our focus is shifting more to prescribe those drugs. Something else to note is that 15% of drugs in the market as of right now have data on pharmacogenomics. So we kind of know essentially that these drugs interact with certain enzymes. We may not know how to like utilize them to the best of our ability with those, but we may be able to at least recognize how they interact with our body. This was something I didn't think was going to be a question when I was taking this course, but it was, so I'll throw it in here. It's these two services right here. You have Farm GKB as well as CPIC. Um, Farm GKB is essentially an organization that measures the evidence level of pharmacogenomics that a drug may have. So let's say it's a drug that's currently being studied. Farm GKB will essentially pull from all the literature, like, oh, CYP3A4 metabolizes this much more heavily than like CYP2D9. And it'll kind of rank the level of evidence based on the research in the field. CPIC, on the other hand, in my opinion, is a little more applicable, as it will essentially combine data in the field to not only describe how relevant the data is, but also how prescribers can use it in terms of prescribing medications. So if there's a really bad CYP interaction, CPIC is essentially the organization that will send out that message to doctors like, hey, if you know this person has this CYP deficiency, don't prescribe this drug in them. So yeah, I just know that basic level of these organizations. You don't need to know too much more beyond that. But just CPIC is the one that helps prescribers, and PharmGKB is kind of the one that compiles things from the literature. Next, let's talk about how we can like get a DNA sample from people for DNA testing. So there's 3 ways. You can get it from blood, and something to note is that you can't actually get DNA from red blood cells since they don't have a nucleus. So we have to pull our DNA from the white blood cells. You can get a cheek swab, which is more of like a skin cell thing. You're not like harming your subject, you're still swabbing inside of your cheek, but the idea is that the cells from your cheek will fall off a little bit onto that swab, or from your saliva, cells and enzymes floating, or not so much enzymes, but cells floating around in your saliva, you can generate DNA from them as well. Something we can do that's really useful is called PCR. That's essentially how we can clone DNA. You need some things to do it. You need a DNA strand, you need RNA primers, nucleotides, DNA polymerase. Kind of makes sense, same components that our cells use to clone DNA. And the enzyme we actually do this through, since to clone DNA, we actually need to split it up into 2 halves. we actually use reverse transcriptase. You may remember that from our previous exam 2 content. That's the enzyme that viruses have that allows them to turn RNA into DNA. And essentially each time we run a PCR cycle, we get 2 to the nth power DNA strands that we can work with and analyze. So if you do 2 to the n, 2 to the 1 for one cycle, that means you get 2 strands of DNA. Whereas if you do 2 to the n, let's say you do 2, so 2 to this second power, that's 4. So we're essentially doubling each time we do this sequence, is how you can think about it. Or if the 2 to the n metaphor makes a little more sense for you, you can use it as well. You might get a question in that. I believe how he worded it for us on either a quiz or exam was like, we have 16 strands of DNA, how many cycles did it go through? So a way you can think of that problem is like, let's say 16, just divide it by 2, so then that's 8, 8 over 2 is 4, 4 over 2 is 2, and then that over 2 is 1. So that was 4 cycles. So that's how you do that kind of problem. Next, let's talk about how we actually kind of like analyze our DNA sequence. The gold standard is what is called Sanger sequencing. You don't need to know too much about it other than the fact that this was one of the earliest methods we use. I'll write down Sanger right here because you probably will get a question on it. But all I would know is that it's simple, it's cheap, and it only measures about 1,000 pairs of nucleotides, so it's not huge segments of DNA. This is before we had like the entire genome mapped. We had the capabilities for this, but to this day, Sanger is still a really good kind of system. If we're looking at a specific region, that's a little bit smaller. The next one is TACMan. The way this essentially works is it'll put fluorophores, which are essentially chemicals that will light up when we shoot a certain laser at them. Um, think like fluorescent for fluorophore. And this can be really useful if we're looking for very specific sections and we kind of know where they are. We can tag them with these fluorophores to see how many times they're expressed, or if the fluorophores don't bind and we think they will, what's missing. So that's how we can use TACMan. The next one is DNA array hybridization. This one is very expensive, is what I remember from it. Essentially, it will stack pairs of nucleotides on an actual physical chip. I don't know too much about the science, but just know that DNA array hybridization, also called gene chips, is literally just stacking something on a chip, and the higher the stack, the more it's expressed. That's pretty much what you need to know for the purposes of this course. The final one is the most complicated one, arguably the most useful, or at least the most powerful. It's called next generation sequencing. And there are 4 steps. You will need to at least know these. You won't need to know what they do too much in depth, but I'll go over it briefly here. Your first step is going to be library prep. This is essentially taking a library of genetic information and like cross-referencing it with other genes. Cluster amplification is similar to sort of PCR in a way where you're upscaling all the specific genes you want to upscale. You're just not doing like a full DNA strand. Sequencing by synthesis, this is kind of similar to like a, I'd say the best way to describe it is that it's kind of like a gene chip in the sense that you're like grouping things off, but it's not on like a physical chip. You're essentially just getting things in certain areas, and you are creating more of them at the same time if they're more amplified. And the final is alignment and analysis. This is where you line up. So you can see this is right here the same throughout this whole thing, and here's like a hot spot. Alignment and analysis is the last step that you're kind of just cross-referencing what your genes do, how frequent are they. Again, you don't really need to know these in detail. But you do need to know the 4 steps and the order they occur in. So that I would highly recommend memorizing these in the order they occur in, but the steps themselves aren't as important. Finally, I want to talk briefly about how some of this information is used. So there's something called a genome-wide association study, or a GWAS. And this will essentially use what's called a Manhattan plot to reference genes in a well-defined cohort without a trait. So essentially, this is when you are just looking for associations in a population, but they don't have any physical difference or illness as far as you know. And what this Manhattan plot can do is it'll put all these dots together But then if there is like an actual association that occurs, you can kind of see it. There's like this line that goes up or the series of points, and that can kind of mark a protein or an enzyme that shows something different. The opposite to a GWAS is a PHWAS. And this is essentially the opposite in the sense that your cohort begins with a certain trait, and then from there you're kind of trying to display how genes affect the phenotype. You're essentially looking at the genes second after looking at a group of people. So you might have like certain people with a certain condition, and then you can rule out based on these genes which genes do we think are causing these variations. Because let's say all these people have a condition. Let's say the green ones have one mutation, the yellow ones have another. Um, that's essentially what a PHWAS is. So you probably will get a definition question based off that. I believe we had a GWAS question and not a PHWAS question. So I know GWAS a little bit more frequently because Dr. Phoenix covered it a bit longer, but just be aware of both acutely. Um, Finally, some companies will commercialize genetic information test. They're kind of accurate. Like you've probably heard of 23andMe. I've heard of Nebula a little bit less, but I think I have heard of that one before. Um, Take these with a grain of salt. They usually look at like one chromosome and only a part of that, um, which they can be useful and they can be kind of a fun novelty. But I'll explain this a little more in a future lecture. The FDA actually doesn't like regulate these, or at least not the information they output. They do regulate like the safety of the tests themselves. Um, so kind of with a grain of salt, they can be kind of accurate, but they're not the most important thing with that in the world. Um, and with that, I will see you in the next video.